Good day everyone, you're welcome to my YouTube channel, Everything Medical. In today's class, I'll be teaching ketogenesis, which is the production of ketone bodies under metabolism. Then, what is ketogenesis? What is ketogenesis? The ketogenesis simply means the synthesis or the production of ketone bodies. That is all about what? Ketogenesis. As you can see from the word, keto is ketone, and then genesis is the production. So it simply means the production of what? Ketone bodies. But before we go further on this pathway, let's talk about the hormonal interplay on ketogenesis. One thing you should understand that this pathway, which is ketogenesis, works under starvation state and in the case of diabetic mellitus. So it works in two conditions. That is the case of starvation and diabetes mellitus. So it works in the case of starvation and diabetes mellitus. How does it work in the case of starvation? When you are starved, automatically you are hypoglycemic, and hypoglycemia causes a decline in insulin secretion and causes an increase in glucagon production. So that means high glucagon, low insulin ratio is potentially ketogenic. Automatically, this pathway is being activated or favored by glucagon. Insulin does not favor this pathway because it works when you are starving, under starvation state. And how does it work under diabetes mellitus condition? Because in the case of diabetes mellitus, there is insulin deficiency. So there is no availability of insulin. Automatically, there is no availability of glucose in the cell or in the tissue because there is no insulin to activate the glucose pathways and even open the glucose channels. So that is it. Then let's talk about the cellular compartment of ketogenesis. Cellular compartment. Cellular compartment. One thing you should understand under metabolic pathway is that it takes place in two parts of the cell. Either it is occurring in the cell cytosol or it is occurring in the cell mitochondria. Cell cytosol is same thing as the cell cytoplasm. So this pathway works in the cell mitochondria. Automatically, it is a mitochondria-based pathway, and all the enzymes are called mitochondria-based enzymes. So it occurs in the mitochondria part of the cell. Mitochondria part of the cell. Another heading that we'll be looking on to is the organ of metabolism. Organ of metabolism. What is the organ of metabolism of this pathway? This pathway works in all the organs of the body except the RBC. It does not work in the RBC. Why? Because the RBC lacks mitochondria. The red blood cell does not have mitochondria. That is why it does not feature in the red blood cell. And in terms of the energy production, that is the energy utilization by the extrahepatic tissues, which are those tissues that is not liver. The liver is the only organ that tends to produce ketone bodies, but it does not make use of energy from the ketone bodies. That is the only organ that does not make use of the energy from ketone bodies, and as well the RBC. The liver and the RBC does not make use of the energy from the ketone bodies, but the liver is the organ that produces those ketone bodies. So that is it. That is the organ of metabolism, is that it occurs in all the tissue except the RBC. The RBC lacks mitochondria. Now let's go to the divisions of the ketone bodies. The ketone bodies are divided into two. Divisions of ketone bodies divisions of ketone. Ketone bodies are divided into two. We have the primary ketone bodies, primary ketone bodies. Then we have the secondary ketone bodies. Secondary ketone bodies. The primary ketone bodies is known as the acetoacetate. We call it the acetoacetate. The primary ketone bodies is primary ketone body is acetoacetate, while the secondary ketone bodies are two. The secondary ketone bodies are two, known as the acetone and beta hydroxyl butyrate. Acetone and beta hydroxybutyrate are known as the secondary ketone bodies, while acetoacetate is the primary ketone bodies. The most important ketone body in the body is the acetoacetate. The acetone and the beta hydroxybutyrate are not important. They are not useful to the body. They are nonsense. They are not useful at all. They don't feature in any metabolic fate nor in energy production pathway. Do you get? So the acetone is the most important. Why? Because during starvation, the acetone is being activated to acetoacetic-CoA, which enters beta oxidation to produce energy. Here is the reaction. Acetoacetate is being activated to acetoacetic-CoA. Acetoacetic-CoA. Then acetoacetic-CoA will enter beta oxidation. Beta oxidation. Why does it enter beta oxidation? It enters beta oxidation to produce energy. So the essence of it is to do what? Is to produce energy. That is why it enters beta oxidation. But it has a reaction. It has an enzymatic function. Acetoacetate, which is the primary ketone body, is being activated to acetoacetic-CoA by an enzyme called thiophorease enzyme. Thiophorease enzyme, which has a coenzyme known as the succinctly and the succinate. The succinctly and the succinate, it goes this way. The succinctly is converted to succinate. So as you can see on this reaction, the most important intermediate in ketogenesis pathway is acetoacetate. That is why it's known as the primary ketone bodies. So this one is useful to the body. Why acetone and beta hydroxybutyrate are not useful to the body? They are nonsense. They don't feature in any metabolic fate nor pathway. So that is all. Let's dive into the metabolic pathway of ketogenesis. But then, before we go into that, let's give a little bit study about the hormonal interplay of ketogenesis. One thing you should understand is that since this pathway works during starvation, so automatically there is no insulin available. So it means that high glucagon, low insulin ratio is potentially ketogenic. So the hormone called glucagon favors ketogenesis. It activates their enzymes, while insulin does not favor ketogenesis, because this pathway works under hypoglycemic state, whereby there is no availability of glucose, automatically there is a decline in insulin production. So let's go into the pathway proper. Number one reaction of this pathway is talking about condensation. So here we have what we call number one, condensation. Why? Because two molecules of acetyl-CoA is condensed to form acetoacetyl-CoA. To condense to form acetoacetyl-CoA. By an enzyme called acetoacetyl-CoA synthase. Acetoacetyl-CoA synthase enzyme. Now, there will be a removal of CoH. CoH is being what? Removed. CoH. That is the condensation of what? Two molecules of acetyl-CoA to form acetoacetyl-CoA. That is step one reaction. Step one reaction. We go to step two. Step two is all about the production of HMG-CoA. Production of HMG-CoA. Here we have our HMG-CoA. Acetoacetyl-CoA is being converted to HMG-CoA by an enzyme called HMG-CoA synthase. HMG-CoA synthase enzyme. That is the enzyme that tends to catalyze the reaction. But then, before acetoacetyl-CoA will be converted to HMG-CoA, there will be an addition of one molecule of acetyl-CoA. So there is what? Addition of one molecule of what? Acetyl-CoA. So it goes this way. H2 of water to CoA. Now, the number three reaction goes this way. Is all about the lysis of HMG-CoA. Lysis of HMG-CoA. That is how the reaction goes. Here is step two reaction. Go to step three. HMG-CoA is lysed to produce acetate and acetyl-CoA. To produce acetate plus acetyl-CoA by an enzyme called HMG-CoA lyase. HMG-CoA lyase enzyme is a hepatic enzyme. HMG-CoA is the only enzyme that is mainly found in the liver. We call it hepatic HMG-CoA lyase. That is why this pathway can only occur in the liver. The liver is the only organ that tends to produce ketone bodies. But in respect to their energy production in giving to other tissues, it is being utilized by an extra hepatic tissue, which could be the brain, which could be the heart muscles, the renal cortex, and the skeletal muscles. Those are the extra hepatic tissues that tends to utilize the energy from ketone bodies. But in respect to those energies production, it is being produced from where? The liver. Because the liver is the only organ that has the enzyme that tends to feature in the production of the ketone bodies, which is the HMG-CoA lyase is a hepatic enzyme. We call it hepatic HMG-CoA lyase enzyme. So that is how it goes. Here is our step 3. We'll go to step 4. Step 4 is all about reduction. Step 4 is reduction reaction. Acetoacetate is reduced to beta hydroxybutyrate. Beta hydroxybutyrate is reduced to beta hydroxybutyrate by an enzyme called the Ketogenesis enzyme, which requires a coenzyme which is NAD plus H plus H, because it's a dehydrogenase enzyme. So the enzyme is the dehydrogenase enzyme. The dehydrogenase what? Enzyme. That is the enzyme that tends to catalyze the reduction of acetoacetate to beta hydroxybutyrate. This is step 4. Then step 5 is spontaneous decarboxylation. Step 5 is what? Spontaneous decarboxylation. Step 5 is what? Spontaneous decarboxylation. It goes this way to produce acetone. Since it's a spontaneous decarboxylation, CO2 is removed because it's a decarboxylation reaction. CO2 is what? Is removed. And it is non-enzymatic function. It does not have any enzyme featuring. There is no enzyme catalysis. So it is non-enzymatic. It is non-enzymatic. Recall that acetone and beta hydroxybutyrate are the secondary ketone bodies. Secondary ketone bodies. We call them the secondary ketone bodies. Why acetoacetate is the primary ketone bodies. Then what is the meaning of HMG-CoA? What is the chemical name of HMG-CoA? HMG-CoA simply means HMG-CoA simply means beta hydroxyl beta methyl glutaryl CoA. HMG-CoA simply means beta hydroxyl beta methyl glutaryl CoA. That is the chemical name of HMG-CoA. But then we have two HMG-CoAs in metabolic biochemistry in respect to their cellular compartment. So here it goes. HMG-CoA, HMG-CoA. This is mitochondria, while this is cytosol or cytoplasm. The HMG-CoA of the mitochondria or the mitochondria HMG-CoA is the one that features in ketogenesis. So is a ketogenic HMG-CoA. Now, why the HMG-CoA that features in cytosol or found in cell cytosol is the one for cholesterol biosynthesis? It serves as an intermediate in the synthesis of cholesterol. Automatically, the cytosolic HMG-CoA is used for cholesterol biosynthesis, while the mitochondrial HMG-CoA is used for ketogenesis. So you have to note their key differences in respect to their cellular compartments. So that is it. Then let's go into the applied biochemistry of ketogenesis, which is ketosis. Which is what? Ketosis. Ketosis. This ketosis involves three components. The ketosis tends to have three components. We have number one, ketonemia. We have number two, ketonuria. And number three, the smell of acetone breath. This ketosis is commonly caused by an untreated diabetes mellitus. That is what that causes ketosis. Recall that the rate of synthesis of ketone bodies in the liver is minimal. But then, when the rate of synthesis of ketone bodies in the liver exceeds the utilization by the extrahepatic tissues, it will be accumulated in the blood, leading to what we call ketonemia. It will as well be accumulated in the urine, called ketonuria. Then there will be presence of acetone in the lungs, which is known as the smell of acetone breath. That is why it has three components, which is ketonemia, ketonuria, and smell of acetone breath. You have to note that what are those extrahepatic tissues that makes use of the energy from ketogenesis? We have the brain and the skeletal muscle. The brain and the skeletal muscle make use of the energy from ketogenesis. Alternatively, they use energy from ketone bodies as their alternative source of fuel, because the glucose is the main fuel for the brain and the skeletal muscle. But in the case of starvation, whereby there is no availability of glucose, they will use the energy from ketone bodies as their alternative fuel, which is for brain and the skeletal muscle. But the heart muscles and the renal cortex prefers the energy from ketogenesis as their main fuel than the glucose. But the brain and the skeletal muscle uses the energy from ketogenesis as their alternative source of fuel. You have to note these tissues, these extrahepatic tissues that make use of the energy from ketogenesis. That is how it goes. So, thank you for watching, and anticipate for my next class on metabolic biochemistry. Thanks.